Hi, everyone. My name is Bill Daniels, and I'm the program leader for Native Seed Communities, which is a project of the Indiana Native Plant Society. And we're very glad that you've joined us tonight for David Sollenberger's presentation. Before I introduce David, though, I'd like to share with you some information about Indiana Native Seed Communities. We promote networks of native plant enthusiasts working together to regionally procure, to process, and propagate native plant seeds for the purpose of increasing these beautiful and ecologically appropriate native plants in all of our landscapes. We have a lot of resources on our Indiana Native Plant Society's website, and Deb Housen, who's helping tonight, she'll be sharing some of those links with you all. A few final notes before we go on to our speaker. For the best experience for everyone, if you'd please keep yourself muted, unless, of course, you're invited to unmute. Also, if you have any questions, Deb will be doing the Q&A after the presentation. And if you would, just pop your questions into the chat, and then she will field those, and then she will moderate that part of the meeting. So I'm happy to introduce David. David has spent the majority of his personal and professional life in the Chicago area. After graduating from Southern Illinois University-Carbondale in 1980 with a BA in Botany, he applied for and received a position at the Chicago Botanic Garden as naturalist for the management of their remnant woodland and also the development and installation of a series of prairie habitats. Eventually, he put his energy into the development and management of the Dixon Prairies and supervised another position to manage Turnbull Woods. About 15 years ago, he was hired to manage the newly established Dixon National Tallgrass Prairie Seed Bank at the Chicago Botanic Garden. The bank currently holds over 4,000 seed accessions. I now present to you David Sollenberger. All right, David.
All right, thanks, Bill. Let's do the share screen here. So we can do it. Let's get started. I think that most of you at some time or another have seen headlines like this in articles. 2,000-year-old seed sprout sapling is thriving, or exceptional seed longevity and robust growth, ancient sacred lotus from China. First one's a 2,000-year-old seed, and the second one, what is it? They're saying 1,288 years old. And then the upper right, you got one from Russia that's 30,000 years old. Well, these are exceptions to the rule, but seeds do have longevity. I believe the one from Russia was not actually a seed that was germinated, but they used the tissue in vitro tissue culture to start that plant. But anyway, you get the idea that seeds can live for a very long time. And there's two important characteristics of seeds that I think make seed banking relevant. A handful of seeds harvested across the breadth of a plant population possesses most of the genetic diversity held in that population. And the longevity of orthodox seeds, which are desiccation tolerant, that's what we'll be talking about tonight, can be extended when dried to a specified percent humidity and then frozen to a specified temperature. So those two things really make seed banking possible and probably desirable. Oh, that's interesting. I skipped a slide. That's odd. Anyway, I had a slide that showed contents of what I'm going to present tonight, but I guess we'll just get right into it. Let's start talking about this history of seed banking in general. Traditionally, seed banks have been used to preserve economically important crops and their wild relatives. Most of your seed banks today are set up for that kind of work. And then more recently, seed banks have been instrumental in preserving germplasm of rare plants threatened with extinction. I don't know if you guys are familiar with CPC, the Center for Plant Conservation, but they're have been instrumental in using seed banks for that kind of thing. And then even more recently, seed banks have been recruited to preserve genetic diversity of native plants, communities impacted by habitat destruction, invasive and or exotic species, and climate change. That's what we're doing in Chicago, and a lot more seed banks are starting to do that as well. I don't have to tell you that native habitats are dwindling, and genetic diversity is decreasing. So we think that this is probably an important thing to do. These are some intrinsic factors that allow long-term seed storage. After morphogenesis, which is the seed development, which is the formation of the embryo, endosperm, and testa. After morphogenesis, orthodox seeds enter the maturation phase, during which they acquire desiccation tolerance, followed by a reduction in water content, and they become quiescent or dormant. The embryo stops growing. Also, cellular cytoplasm is transformed to a glassy state. It's a reduction of water and accumulation of non-reducing sugars and various desiccation-tolerant proteins, and where cellular metabolism slowed to almost non-existent. And that is why some of these orthodox seeds, desiccation-tolerant seeds, are able to survive for long periods of time. And extrinsic factors that enhance long-term storage by slowing cellular metabolic activities. So even though this is happening naturally, there are things you can do to enhance that, their longevity. And one is collect seeds at peak maturity. Make sure you don't damage the seed coat. Dry seeds and store them at 15% relative humidity. And you store seeds at minus 20 degrees C. These are pretty typical criteria for seed banking. All the seed banks generally do this. Again, for orthodox seeds. Oh, I don't know how these slides got mixed up, but this is the summary that I wanted to talk about earlier. History of seed banking, we've done that. History of Dixon National Tallgrass Prairie Seed Bank, which is the one I'm involved with. That's coming up. We're going to talk about the evolution of the restoration collection, which is our major collection at the seed bank at DNTPSB. And we'll talk a little bit about seed collecting protocols, processing seeds for long-term storage, and diversifying our collections, and expanding collections in the Eastern Midwest. Okay, this is a very simplified account of seed aging. I'm not a seed physiologist, but as far as I know, this is, hold on a minute, I got to get rid of this thing. This is, hold on a minute here. There we go. Okay. ROS influence. The aging process in seeds appears to be related to elevated accumulations of reactive oxygen species molecules. That's a funny name, reactive oxygen species, but that's the name of a molecule, ROS. And they're mostly free radicals in the seed cell cytoplasm. This occurs when various antioxidant enzymes, which neutralize ROS, decline in concentration over time. An increase of ROS molecules in a cell oxidize lipids, inactivate enzymes, and can be disruptive to the structure and function of proteins, carbohydrates, RNA, and DNA. Seeds with high lipid content generally age faster, as ROS oxidation readily affects the permeability of membranes, which are primarily constructed of lipids. The production of ROS molecules is a natural byproduct of photosynthesis and respiration. An effective antioxidant system prevents their overaccumulation in seed cell cytoplasm. In small concentrations, ROS can be beneficial as triggers for gibberellic acid production and breaking seed dormancy. Note, upon imbibition, cells have the ability to make repairs to damaged proteins and genetic material caused by the increase in ROS molecules during storage. If damaged beyond repair, the death of the cell ensues and ultimately the loss of seed viability. So even though the increase of ROS molecules creates seed aging, intensifies seed aging, the cells do have the ability to repair some of the damage to RNA and DNA and proteins and carbohydrates when they're imbibed at germination. What's going on here? I'm having a heck of a time with this. It won't advance. Oh, there it is. Okay. Not sure what happened there. This is just a diagram that explains it a little better, I think. Down below, you have the year and percent viability. You notice that as the years proceed, percent viability decreases. And what happens above is you have quiescent or mature seeds that were collected in the bank. And as I mentioned before, antioxidant enzymes start to decrease. And remember, the antioxidant enzymes are the ones that control the production of ROS, or the accumulation of ROS molecules. So they start to increase, and when that happens, they start to degrade the structure and function of proteins, lipids, RNA, and DNA, and then eventually you have cell death and attrition of seed viability. And then off to the right, upon imbibition, cells have the ability to make some repairs to proteins, RNA and DNA, damaged by ROS storage. So that's kind of what's happening to the seeds in the seed bank. And we try our best to slow that as much as possible. Why is this not advancing? Huh. Sorry, folks, I don't know what's going on here. Oh, okay. Okay. Determining seed longevity, the germination testing dilemma. Currently, testing for declining seed viability in banks involves periodically removing a sample of seeds from the collection and performing seed germination assays. However, this is not an ideal way to test seed longevity because germination is binary. Either seeds are alive or they're dead. And then there's another problem with the determining seed longevity through germination testing, and that's the number of seeds needed in germination samples. And that's a destructive process. So you're losing seeds every time you do germination tests. And if you have a small collection, that can be a problem. However, there's a process called RNA Integrity number, RIN, that's being the Center for Plant Conservation and the USDA National Laboratory of Genetic Resources has teamed up to apply RIN to measure seed longevity. And RIN calculates RNA degradation in a linear process so that changes in seed longevity can be detected before germination viability losses observed in germination tests. So where you have, and you can see this diagram here, the little boxes with the red 10, 20, 30, those are generally when seed banks do viability testing every 10, 20, 30 years, every 10 years. So, and the big colorful arrow at the top showing RNA degradation, it increases as it increases, and it will with seed aging. The RIN values increase, and then they get to a point where the cells can no longer repair damage to the RNA, and cells die, and eventually the seed dies. So if, for instance, here, you can see at 20 years, the seeds are dying. So if you test them at 10 years, everything looks good. 20 years, not so good. Because it's but because RIN is linear, you can see that that the seeds are declining all at a steady rate. So it's a better way to determine seed longevity. They do it with crop plants right now, and they're trying to use this same technique for wild seeds. Okay, the history of the Dixon National Tallgrass Prairie Seed Bank. In 2000, the Millennium Seed Bank Project, MSB, began its mission to collect seeds of 1/10 of the Earth's flora. You may have heard about this project. They partnered with the U.S. Bureau of Land Management, BLM, to collect seeds in the U.S. In 2001, BLM established the Seeds of Success program, SOS, to collect seeds for MSBP and provide seeds for habitat restoration. Chicago Botanic Garden partnered with SOS to collect seeds for MSBP in the Midwest, along with other institutions in the East where federal public lands are fewer. So BLM has it covered out West, and they needed partners in the East to collect seeds in this country for Millennium Seed Bank Project. The Dixon National Tallgrass Prairie Seed Bank was created in 2008 to continue collecting seeds in the Midwest to conserve genetic diversity of our native flora. So collecting protocols for Dixon National Tallgrass Prairie Seed Bank are similar to those established by MSB and SOS, but unlike collections made by MSB, our seed bank focuses on multiple collections of species from ecoregions comprising the tallgrass prairie biome. So the Millennium Seed Bank, when they're dealing with the seeds of the Earth's flora, one collection is plenty. But we thought we would do multiple collections across the region, and you can see that map on the right. Everything within that bright green outline is basically the tallgrass prairie region, except for maybe down in Texas. And all those different colored areas within our ecoregion. So we were trying to collect seeds, multiple collections from each of those ecoregions. So a little bit different than what Millennium Seed Bank, how they approached it. But we thought this was an important way to preserve seeds from across the region. Okay, protocols for collecting genetically diverse wild seeds without compromising the integrity of the population. All seed banks strive to do this. Our goal is not to wipe out the plants in these populations, but to preserve them. So these are the protocols which were established by Millennium Seed Bank and are pretty common with most seed banks. Select populations of greater than 50 individuals when possible. If you can collect seeds from 50 individuals in a population, you're capturing about 95% of the genetic diversity in that population, which is quite a bit. Collect across the breadth of the population, so not just the middle, but all the edges, different elevations. do not show bias for large or more fecund infrutescens when collecting, and infrutescens is a fancy word for a seed head. Collect no more than 20% of the mature seeds or 10% for annuals, and collect greater than 3,000 seeds if possible while abiding by the parameters stated above. If you collect 3,000 seeds and more, you're able to do germination testing, because you remember there is attrition of your seed source when you have to take samples out to do germination testing. So we try to get at least 3,000 seeds, preferably more. It's good to assess the population and so that you can select the best collecting strategy. If populations are large, well over 50 plants, we recommend a standard bulk collection or an equal representation bulk collection. Standard bulk collection, there's lots of seeds, and given that 20% of the seeds are collected, 10,000 seeds will easily be attained. Equal representation is a little different. That's where inherently the plant has fewer seeds, given 20% of the seeds collected, so that fewer than 3,000 seeds will be attained. And when that happens, you want to collect seeds equally represented, collect the same amount of seeds from each plant. And that way you're equally representing those plants in your collection. If a population is very small, around 50 or less plants, we do what we call a maternal line collection. This often happens with really rare species, and you may or may not, if you collect 20%, you may or may not have 3,000 seeds. And you only do this, collect from rare populations if there's no larger population in the region. Obviously, if you want to capture as much genetic diversity as possible, you're always looking for the largest population. Also, for large and small populations, you can do collections with repatriation. These are species with fruits. Where removing 20% of the seed in the field will be difficult. An example would be milkweeds. To split open a pod and remove 20% is not an easy thing to do. So it's best to collect the whole pod, remove 20% for the seed bank, and then repatriate the rest of the seeds back to the site. And also collecting clonal species is difficult because they spread vegetatively, making it difficult to differentiate genetically distinct individuals for the collection. So we do what we call a spatially discrete collection, where you predetermine spatial intervals to improve chances of removing seeds from genetically distinct individuals. If they're clonal, it's very, very difficult to tell if you're collecting from different plants. So you just kind of decide ahead of time the spacing you're going to use to collect those seeds. This is a restoration target list, which is our primary goal. And this is how we determined what species would be on our restoration target list. We began with the comprehensive NatureServe plant list for Omanic ecoregions level 3 within the core of the Tallgrass Prairie biome, and there's 7 of those ecoregions. So we started with the comprehensive lists. We subtract non-bearing, seed-bearing, and non-native plants from the selected ecoregions. And there were approximately 3,000 native seed-bearing plants that existed in those 7 ecoregions. So the next thing we did was we reviewed the literature, Flora's vegetation surveys, for information indicating the importance of a plant species to common habitats, prairies, woodlands, savanna, marsh, found in those the ecoregions. Didn't include habitats like bogs because that's really a northern habitat. So unusual ones were not included in this. Plants that are common in unusual habitats, bog in the region. So we reviewed geographic range maps to confirm the primary distribution of the species occurring within the Tallgrass Prairie. And we also considered anecdotal information on species, ease of establishment, or overall performance in habitat restorations. And then we ranked those taxa with number 1, 2, or 3. 1 being a conservative species that is important to most common communities in the region. And when I say important here, it means can be defined as dominant or characteristic of a community. Number 2 is given to conservative species that were less important to a common community, or a less conservative species, but is often used in seed mixes for habitat restoration. 3s were given to less conservative species that is important for a common community, or may be used in seed mixes for habitat restoration. And I think, yeah, and this is kind of puts it together, this little table here. So if it's a conservative species of high importance, it gets a 1. If it's a less conservative species with high importance, it gets a 2. Or if it's a conservative species with less importance, it gets a 2. And and if they're weedy species, they're less important, or have a if they're it gets a 3 if they're less conservative species of less importance, or a high importance species that are native, that have native weedy tendencies. So this is the kind of the chart that we used for determining the numbers. And so then we selected the restoration target list, and it turned out to be 544 taxa based on the data from the ranking system, and included all species designated 1 in at least 5 ecoregions, included all species designated 1 in at least 1 ecoregions, and 4 other number designations in at least 5 ecoregions, included all species with at least one number 2 designation. Species in this category were given a lower status on the target list. So that's kind of the process we used. We did not send the list out for review because in the past we've had issues with people just not getting around to it. If you set a date for some kind of response that passes, and it generally doesn't work out. So we included in our website that, if you feel that there should be another plant on the list, or if a plant should be removed, then just let us know. And that has happened, because we now have 545 species instead of 544 based on someone's comments. So anyway, this is what our target restoration looks like, and can be found on our website. And this is what we have people focus on collecting across the Midwest for us. And if you just take the one highlighted in yellow there, GM triflorum, it was collected. There's been 1 collection in ecoregion 48, in ecoregion 52, and in ecoregion 53. And so the other ecoregions may have that plant, they may not. But there's probably room for more collections for that species. So any collectors that we have can look at this target list and see which ones are open for collection. By the way, the ones that are that are highlighted in red are our priority collections. Those were the ones that got the highest numerical ratings. Okay, processing seeds for the seed bank. First, and this is pretty typical for all seed banks, I think, at least for the most part. 1, enter the data from the field data sheets into the database and generate an accession number. 2, you want to dry the seeds at ambient temperatures in a seed quarantine room or the seed processing lab. Record the accession number on the seed collection bag immediately, because that's how you track those seeds. And you got a lot of collections coming in. You got to want to be able to, you don't want to get your data mixed up. 3, label and move herbarium specimens into a plant press. Record accession number on herbarium specimens and the DNA specimen. So our collections aren't just a seed collection. We also get herbarium vouchers to verify that what we have is correct. And we also take a DNA specimen, which is the leaf sample, for our DNA bank. We may not need to look at that at this moment, but we have it there just in case. It's something simple to do, so we do it while we're making the collection. 4, we clean the seeds to several reasons: to reduce space in the seed vault, ensure minimal damage to the seed testa. You gotta be careful when you're cleaning. Maybe more careful than when you're cleaning seeds to be put back out on the landscape, because the testa needs to be intact for those seeds to achieve their greatest longevity in long-term storage. Reduce empty seeds to facilitate quantifying the number of potentially valuable seeds in the collection, and maximize genetic diversity, which means we don't want to throw away large and small seeds because they might have genetics are important for that species. So if that means cleaning them a little less than they could be, so be it. 5, remove a few seeds for a photograph, then move clean seeds into the seed dryers. Those are the seed dryers in the lower right there, and they maintain about 15% humidity and about 15 degrees Celsius. 7, we X-ray seeds. We're fortunate to have an X-ray machine, and you can see this is an example of why we do it. This is Parthenium integrifolium, the wild quinine. I'm sure some of you are familiar with that plant. The seeds, if you look at the far left, they all look about the same. You can't tell if this is actually the fruit. The seed's still inside that, but it's very difficult to clean it down to the seed. So you usually just keep the fruits. You do the X-ray and you can see the bright white in the middle there, the bright white, and there's a blow up on the far right, and you can see the seeds that are intact and the ones that never formed. So X-rays are really important, and generally, if we have if our sample for X-ray is we get 50% of the seeds that are that's showing up to be unfilled, then we re-clean it and go through the process again until we get about 50%. And then 8, the seeds are packaged and stored, clean and dried, 15% relative humidity. Seeds are heat sealed in foil packets to retain that 15% relative humidity, labeled and then stored numerically by accession number in the seed vault, which is a giant freezer, walk-in freezer that's held at 20% or 20 degrees Celsius for long-term storage. Not everyone uses these foil packets, but they're very, very handy, and they do save some space. Okay, just the cleaning operation. This is we clean seeds a little bit more than a lot of practitioners that do restoration. They don't need to clean their seeds that well because they're going right back in the landscape. But we have to quantify what we have and count them, and so we try and clean them to a greater degree. And you can see number 1, this is Allium cernulum. If you follow the numbers, number 1 is it's just been sent through probably a sieve and you get a combination of the black seeds and the chaff. They pour it into a cup, and this is called a column blower, which forces air up through the cup, and it's separate. It moves off the lighter materials, the chaff, and eventually you get not perfectly clean, but much better. And you will probably take a tweezers or a forceps and remove some of the bigger material by hand then. And then that's as far as we go. Not perfectly clean, but not too bad. One more example. This is a little more difficult. This is fireweed, Epilobium angustifolium. Beautiful picture of it in flower, and it's got these fantastic seeds that have coma on them. Perfect for distribution in the wind. And number 3, that's kind of what it looks like after you pull it out of the bag after it's been collected. 4, we take a vacuum and suck the seed chaff mix through a screen. The larger chaff remains behind. If you look at number 5, and the seeds and coma are sucked into the vacuum. Most of the seeds, and you see at number 6 there, most of the seeds fall to the bottom, but the coma and a few seeds stick to that foam core of the vacuum. And so we carefully try and separate we go through the coma that's attached to the core of the vacuum and try and separate what seeds are in there. And 8, you get something that looks like that. And then 9, it goes back to the column blower. And eventually you get your seeds for fireweed. And again, not perfectly clean, but a lot better than when we started. Other equipment in the lab, the heat sealer on the far left, microscope, you got to have a microscope. A lot of the seed is very tiny. You got to make sure you're saving the correct thing, not the chaff, but the seed. And this is a general, far right, that's a general picture of the lab, our lab at the Plant Science Center at the Botanic Garden. There's a balance in the foreground there, and those white bars are static control bars. So a lot of the seed cleaning is done in midwinter when the air is very dry, and static electricity can create all kinds of problems. If you're dealing with really tiny seeds, they either stick or jump. So this neutralizes the surface of the workstations and allows you to clean the seeds with greater ease. And then those black hose-like things are those are dust collectors. That's for our health, so we don't have to breathe a lot of the dust in the cleaning process. This is a diagram, a flow diagram of the seed bank. If you start off to the upper left, you start with your native plant population, and if you follow the blue, that's what the actual movement of seeds through the process, and then all the other boxes in white are associated. So you do the seed collection actually goes to CBG and SOS databases because we share our seeds with SOS. And some of the herbarium vouchers go to CBG and SOS databases, SOS collections, data, photographs, and DNA samples. Then you move on. The seeds are dried in the dryer. Herbarium, I should mention herbarium vouchers are held at the CBG and also are sent to Smithsonian Herbarium. And then the seed dryer, and then it goes to count and weigh. And that's when we pull off a seed for a photograph. And then from count and weigh, we go to x-rays. You saw an example of how we x-ray. And then we divide and package them. And some of the seeds are sent off-site. We have redundant storage in the Pullman, Washington, and the Fort Collins USDA facilities. They take some of our seeds, and then we take for our seeds, we divide them into a germination research lot and a conservation lot. The germination research ones obviously are the ones that we use to track longevity of the seed, and also they're disseminated to nonprofit restoration projects and research projects. And then eventually they get into the seed vault. From the seed vault, they're if they're if they start to run low in numbers, we can either recollect at the native plant population, or we can grow out the seeds and recollect the first and second generations, put them back in the bank. And they also can go out for germination assays, if you look in the lower left corner, or we can distribute them to non-commercial projects, or do grow outs for G1 and G2 seeds for native seed supply chain. We're not doing that yet, but that's what we're thinking about doing in the future. Will be a good resource for seeds across the region, and it'd be a good resource for nurseries to grow out seeds for the native seed supply chain, which they want to develop. Just an example of we have done some germination assays. We don't do it on a regular basis, but we have a lot of students from Northwestern University that work on their masters and PhDs at the garden, and they help us out with doing some germination tests from time to time. And you can see here in green, I don't have they're just numbers, but the seed accession numbers, and you can see they're very low numbers. So these are old accessions. So we always start with our oldest accessions to see how they're faring in the bank. And you can see the seeds that are germinated are in green, and the non-germinants are in blue. And then we have in yellow non-viable non-germinants. So so we have, if they're viable non-germinants, it means the seed looked good. It didn't germinate for some reason. Perhaps it was a dormancy issue, and we need to work on that a little bit more. And then, of course, the non-viable non-germinants are seeds that were destroyed completely, or had molded, or were no longer viable. So you can see it's not too bad. Some of those, we have some dormancy issues, but a lot of them seem to be doing pretty well. Just thought I'd throw this in. Ex situ conservation. I mean, we call it long-term conservation, and we do want to preserve the seeds long-term, but also we want to support in situ conservation restoration projects. And so seeds in a seed bank represent the genetic diversity of a plant population at the point where they're harvested and banked. As ex situ collections, they have been removed from the natural environment and are no longer allowed to adapt to changes in that environment. That's an issue we have to deal with, and this appears to limit our strategic value, their strategic value in the conservation of native plants in their natural environment. However, the natural environment is under stress from a barrage of anthropogenic disturbances, such as to such a degree that the plant's ability to adapt in situ is compromised. Ex situ collections can and should support in situ restoration of natural habitats. Although seeds can remain viable in seed banks for decades, a portion of their collection should be made available for research and habitat restoration projects. So we really don't want to keep them all in the bank. We need to use them and get them out in restoration work. Also, just a note, with the advent of rapidly changing climate, seed banks with a multiple collection of seeds of species in multiple ecoregions are uniquely positioned to provide regionally sourced seeds for assisted migration or managed relocation projects. So yeah, as our climate change is changing, is warming for the most part, The habitats are changing faster than the plants can adapt. So I know in Lake County, Illinois, they're already doing a restoration where they're collecting seeds from further south in Missouri, and because they predict that um, in one of their prairie restorations, they predict that that those seeds will do better in the future because of climate change. So it's kind of controversial. Some people think it's a real mistake to work with this assisted migration, but others think it's necessary. Whoops. let me back up. Future plans. Um, we want to upgrade our online database to include real-time collections map. I've been trying to do this for years. It would be great to see where the collections are immediately. As soon as they're accessioned, they are on a map and can be viewed. Um, initiate seed collections of plants with known ethnobotanical qualities. So it would be, we have our restoration target list. This would be another list of plants that have ethnobotanical qualities. We'd like to improve the phylogenetic diversity of seed bank collections. Instead of having 50 members of the aster family, we could get some from Aristolochiaceae or some of the other unusual families. Participate in the National Association of Restoration Seed Banks, NARSB, and assist in the development of national native seed supply chain. And also, we'd like to expand our collections into the eastern region of the Tallgrass Prairie, where Tallgrass Prairie was not the dominant plant community, but existed in small parcels. It's like the tension zone between the prairie and the eastern deciduous forest. Indiana is a good example. We're trying to expand our collections in Indiana and Michigan and Ohio, Kentucky, and those areas. Okay, that kind of concludes it. And I kind of jumped the gun here, but this is um, a few years ago, some of the seasonals in our native habitats department started making up these botanical puns, and I've been continuing it, and this is one of them. By the way, I thought it might be a t-shirt design for a very select group of people that would appreciate this, and it's called Botany Tees. So that in itself is a pun, but this is one of the kind of crazy ones. Nice coma over, and and then in parentheses coma, you can there's a tuft of hairs, and then this is one that's more topical. Whoops, where'd it go? Oh, I lost it. Anyway. Anyway, I lost it, but that concludes my presentation. I'll stop share.
Thank you, David. That was a very interesting process in the seed bank, and a lot of information that I had no idea what was going on. While we're waiting for any questions that might pop in, I actually had one myself. Part of it you did answer through your presentation, but it basically then is the grow out period. So when it sounds like part of your supply is dwindling through either testing or germination viability, you then send some back out to be grown or go and collect more, correct? Is there a time frame, or is it based solely on the quantity of seeds that you have?
Yes, to both of those. We haven't had to do that yet, fortunately. But it will happen in the future. A lot of the seeds that we have in the bank go towards research projects, especially with our students. from Northwestern, and also outside the garden. And we only disseminate between 50 and 100 seeds. So it's not like they're getting lots of seeds from our collections, but over time that'll increase. And yeah, if the site where it was originally collected still exists and we have permission to collect again, we would do it that way. That would be preferable. But as the seeds start to dwindle, and I don't know what percentage of seeds that would represent, or that would we would reach to where we would make the decision to grow them out. But we have to make that, we have to determine that. We probably talk with other seed banks and see what they're doing. But
Yeah.
it hasn't been an issue so far, so we haven't done anything yet.
I would suspect that if it's a fairly, I don't want to say common, but common native, there's less need to maybe go out there and collect more versus something that's been a little bit more rare or becoming on an endangered list, that you keep a closer eye on the seed bank that you have and what you're collecting. Bill asked, in germination assays that have a low germination and non-germinated seed, mainly not viable, what do you do with the specific accession?
Well, we'd probably keep it as long as there's some viable seed, but it would probably go back on the list to be collected again at some other population within an ecoregion, whatever ecoregion that came from. But we wouldn't throw it away unless it's completely unviable.
Okay. I don't have any other questions that are coming in. Oh, wait, sorry, I do have one. Do you strictly handle orthodox seed, or do you work with recalitrate species like Zizana, or I apologize for my pronunciation, Noluba?
Yeah, recalcitrant seeds. Yeah, we don't work with recalcitrant seeds or intermittent seeds at this point. We need to go into cryopreservation and use liquid nitrogen, which we could do, but and you're only extending their viability for a few years, really. So it's not an idea. It's a kind of an expensive process for not much reward. But we may go that route. I mean, other banks have done it. They do it at the USDA banks, but we're a small regional bank, and we're not set up for that yet. The majority of vascular plants, perennial and annuals in this region are orthodox. So fortunately, so there are, acorns, the oaks, we can't preserve those. Those are all recalcitrant seeds, and hazelnuts and hickories. So, but for the majority of the seeds in this region, there are orthodox, and we can store them long term in the bank.
Great. All right. I think that does do it for the questions at this point. I thank you again. I will turn it over to Bill Daniels to wrap up, and we'll go from there.
I think my microphone's on. Can you hear me okay?
Sure can.
All right, great. Thanks, Deb, for leading that part of the session and for helping with the technology aspects. And David, thanks so much for your presentation. I know I heard you speak. We were in a few meetings for the Indiana Plant Conservation Alliance, and I really thought it was fascinating. The work that you do, and I appreciate the break. Wind up the meeting now, but if you have any additional questions regarding the presentation or native seed communities, you can reach out to the links that Deb has provided in the chat. Also, if you want to be on our email listing, you can, I think she shared our email, seed@indiananativeplants.org. You can go directly there or put it in the chat as well if you're not already on our email listing. We don't send out a lot of stuff, primarily what presentations are coming up and everything. And in fact, Jillian Harris is on the call, and she helps a huge amount with that kind of communication. We invite you to join our Facebook group. Pictures and videos are welcome of how your propagation is going. And also, we're very interested in any of your community projects and how they are going. We've got about 3,000 people that are members of the Facebook group, and it's a good place to be able to share stuff. All right, our next presentation will be Monday, June the 24th, 7 p.m. again. And for this one, we'll be having John Brittenham. He's a restoration biologist for Blue Heron Ministries. He'll be talking about basic growing natives from seed. So I hope to see a number of you all that are here tonight there. So thanks again, David, for your informative session. Again, I'd like to thank Deb for her help. It was good to see you all. We'll sign off for now, and happy growing natives from seed. Bye-bye, all.
Thank you.
Thank you.